Some microorganisms can survive boiling water, radiation, or even the vacuum of space. These survival experts are bacterial spores, and they're among the most resilient biological structures known to exist. That's exactly why they're used to validate sterilization and biodecontamination processes. When laboratories or pharmaceutical facilities validate a sterilization process, they're asking one critical question. Can this system reliably destroy the toughest microorganisms under the most challenging conditions? To answer that, validation protocols don't rely on assumptions. They rely on scientific challenges, including biological indicators, highly resistant organisms, defined microbial populations, measured log reductions. Together, these elements prove whether a system truly works. In controlled environments, contamination is a risk. In animal laboratories, it can compromise research and affect animal health. In pharmaceutical manufacturing, it can lead to batch failures, regulatory findings, costly investigations, and even product recalls. That's why contamination control strategies depend on validated, repeatable sterilization processes, not guesswork. Biological indicators, or BIs, are standardized test systems containing known populations of resistant microorganisms. They allow facilities to measure microbial kill directly rather than assuming a process is effective. Most BIs use bacterial spores because spores are significantly harder to destroy than typical bacteria, viruses, or fungi. If a system can eliminate spores, it provides strong evidence it can eliminate everything else. Bacterial spores are ideal for validation because they are built to survive. They have thick protective outer layers, extremely low metabolic activity, resistance to chemical and environmental stress, the ability to persist in harsh conditions for long periods. This makes them the ultimate worst-case challenge organism. The hardest organism to kill depends on the sterilization method, and different technologies require different challenge organisms. Steam and hydrogen peroxide systems often use Geobacillus stierothermophilus. Ethylene oxide and chlorine dioxide systems use Bacillus atrophius. Validation always matches the organism to the chemistry being tested, ensuring a true worst-case challenge. It's not just the organism, it's also the number of microorganisms used in testing. Biological indicators often contain 1 million spores. Why so many? Because sterilization performance is measured using log reduction. A 6-log reduction, the industry benchmark, means reducing 1 million organisms to 1 or fewer survivors. This level of reduction defines true sporicidal efficacy. Validation doesn't happen under ideal conditions, it happens under the hardest ones. Biological indicators are placed in difficult locations, such as inside complex equipment, behind obstructions, in isolators or biosafety cabinets, or under equipment decks in airflow-restricted areas. Manual cleaning can vary from person to person, making it difficult to validate consistently. Automated biodecontamination systems solve this by delivering controlled, repeatable cycles. Systems like Curis hybrid hydrogen peroxide technology distribute disinfectant evenly while maintaining precise cycle parameters. Once validated, these systems provide consistent results and detailed audit-ready documentation. Microorganisms are incredibly resilient. Sterilization validation proves that your system can overcome that resilience every time. By combining resistant organisms, high microbial populations, and worst-case conditions, validation provides something every facility needs: confidence. Confidence that contamination risks are controlled, confidence that your process works, and confidence that your facility is operating at the highest standard.